Hi, everyone. This is Caroline with Fiveable. I'm excited to talk to everyone tonight about an intro to Unit 1 for teachers for AP Biology. So thank you for tuning in on this Tuesday evening right after a lovely Labor Day weekend. Just to reiterate, tonight will be focused on attacking Unit 1 for teachers. So students, this one isn't necessarily for you unless you're interested in all of the logistics that go into planning your units. But I won't be reviewing any content, more just talking about the content that needs to be covered, some strategies to cover that content, common misconceptions, etc. As always, if we have some people joining and people want to comment or share their own best practices, I would be more than happy to allow for those conversations to occur, as we can all learn so much from one another. And this is more oriented towards new teachers or just anyone who's looking for a little extra assistance in the classroom. All right, so I'm going to make this screen our focus, and we will jump right in and talk a little bit more. And for those teachers who just started teaching today, have a wonderful start to your school year. Okay, so coming soon, just so that you can let your students know what we have coming up. Within the next 2 weeks, we have structure of water and hydrogen bonding. That's going to be tomorrow evening at 8 p.m. Eastern. Then we have elements of life on Thursday at 6 p.m. Eastern. Introduction to biological macromolecules will be next Monday at, I believe, 8 p.m. And then properties of biological macromolecules will be next Wednesday. So lots of good screencasts. Those will all be for students, but obviously teachers can tune in as well if you are interested in hearing more of an overview of content as well. Otherwise, all of those are geared towards students and more review focused. Okay, so tonight, things that we'll talk about. So I wanted to start by just introing AP Bio's units for this year, because that is something that is new for AP Bio for 2019-2020. So we can go through some of the changes to the course. In case you're unfamiliar, I'm assuming that most people probably have looked at all of those differences prior to making their own curriculum this year, but we can certainly still discuss those. And if anyone has any questions, we can go through it. Then we'll overview Unit 1 and then go a bit through the course and exam description from College Board for all of the topics listed below, so that if you have any questions about how to teach something, you can take a look at or ask when we get to that section. Okay. All right, so some changes, and again, I'm sure if you're watching, you very well may be familiar with this, so I won't spend too much time unless anyone has any questions, but it's good to know and kind of keep in our brains the changes that have been made to this course, seeing as we've been used to teaching it one way, and our students are also going to be held accountable to different things. So on the left-hand side, it says currently, that's the 2019 exam, because this came out, I think, in March of this year. So we used to have 69 multiple choice questions, 6 of which were grid-in, an hour and 30 minutes. So we still have that hour and 30 minutes, but we're down to 60 questions, and it's still 50% of the exam, and there are no more grid-in questions, which is maybe good news, because those are like a whole different strategy that we had to teach, as well as some other specific skills that we had to teach. Math will still obviously be a large part of the multiple choice and the free response, but it just won't be explicitly tested in those 6 questions. So less questions, more time per question therefore, and none of those explicit grid-ins. And then for free response, they've cut 2 of the questions, so we go from 8 questions down to 6. Still have those 2 long free response questions, but 4 short answer instead of 6 short answer. And later on in the course, when we get closer to the exam, we can actually look, they've actually said about what kind of topics each of those 6 questions will be, so that we can best prepare our students for that. And again, we have more time per question because we have relatively substantially cut down the number of questions in both of those 2 sections. So that bodes well for our students. Hopefully it gives them a fairer chance and allows them to be more successful with the knowledge that we have taught them. Then the other major change, and obviously bringing this up because we're going to be talking about Unit 1 tonight, is that we're moving away from the big ideas. The big ideas are still really, really important. If you've spent any time looking at the course and exam description, which I have a nice hard copy binder of, which you can request a copy of, which is really, really helpful. If you spent time looking at that we've broken down into these units, that the big ideas are still included, that they're just sprinkled within these 8 units, but this represents probably what a lot of us were doing in our classrooms anyway, and seems to make a whole lot more sense. So you can see the units there. We're starting the year, I don't know how you're pacing out your year, but we're starting the year with 5able, and we're going to go through from Unit 1 to Unit 8, just assuming that a lot of teachers are following the pacing plan set up by College Board. Okay, and then the last bit of information that's really helpful to know as an instructor of AP Biology is just how much each of these topics is going to be showcased on the exam. So you can see between Unit 1 and Unit 8, Unit 1 and then Unit 5, so heredity and chemistry of life are going to be our lowest as far as the amount of times that they're going to be showed on the exam, whereas natural selection, no surprise, is going to be the largest portion of the exam, which is similar to things that we've seen in the past, right? We've seen that natural selection and evolution are always going to be the largest part of the AP exam. So that's just shown to us here. So College Board is giving us the metrics and the tools. It seems as though my take on it is that this year that they're truly trying to provide educators with all the tools that they need in order to get their students to be successful in a very challenging course. Does anyone have any questions? Again, I'm assuming that most people have looked through and rearranged and probably changed things for this year, but just kind of like a high-level overview of some of those changes. And I can post these slides. I mean, they're really nothing special. It's basically just lovely snips from the course and exam description, which everyone has access to. Okay, perfect. So high-level overview of changes. So how do we implement this for Unit 1? So Unit 1, again, the really nice thing that College Board does for us is tells us about how many class periods we should be spending on this material. So because Unit 1 is chemistry of life, they're suggesting 5 to 7 class periods, which is a lot less than some of the other units, but it should make sense that it is a lot less. And this also obviously depends on your students and whether or not they've taken chemistry before, which definitely is going to shift how many days you're going to want to spend on these units, just because there are some really, really important fundamentals here that need to be covered. But if your students are already really familiar with hydrogen bonding, ionic bonding, covalent bonding, and the different elements of life, then they're not going to need as much time on that kind of thing. And you can spend more time talking about like the different structures and functions of macromolecules and nucleic acids and some other things that they might have a little bit more trouble with. So knowing your students, you can kind of pick and choose your own adventure here. But the goal for tonight is just to kind of walk through all 6 of these. I'm going to show you what College Board suggests that we cover in that section. And then we can talk about how people best cover this, strategies to use, common misconceptions that students have, etc. So first and foremost, and I'm not surprised, it makes a lot of sense that College Board is putting structure of water and hydrogen bonding as the first topic in the first unit, because we would not exist, life on Earth would not exist without water and those hydrogen bonds. So I think it's a really cool opportunity to kind of open up your classroom. And there's also a lot of cool demonstrations that you can do with the structure of water and hydrogen bonding, as well as a lot of critical investigations. So we can talk about that in a second. If we skim through this, the main takeaways are that water is essential to life. Living systems depend on it. They also depend on the properties of water. So big things that need to be talked about during this section would be the polarity of water. So it's important to start talking about polarity from, day 1, week 1, whatever it may be. Getting students familiar with the fact that polarity is going to be that unequal distribution of charges, because once you start talking about the plasma membrane and phospholipids and polar and nonpolar molecules that can and cannot get through the plasma membrane, it's going to be really, really essential that they are familiar with those things. So I always like to hit it hard in the beginning and also talk about how hydrogen bonding contributes to that. And then you can see SYI1A3. Wow, they've really made these quite the mouthful since the redesign. But it says the hydrogen bonds between water molecules result in cohesion, adhesion, and surface tension. I always like to do stations here that allow students to see a bunch of these properties. I'm sure we've all done or plan to do, like water droplets on a penny. Another really cool experiment that I like to show students is you can put 2 microscope slides. So you have one microscope slide connected to the desk, and the other one goes on top with just a droplet of water in between the two. So this one, my arm that's moving right now, would be a microscope slide that's hanging off the edge of a desk. And the only thing that's connecting it to the desk is the water molecules in between the 2 slides. And then you can actually balance pennies or poker chips or whatever you have lying around in your classroom. I always use poker chips because I use them for Hardy-Weinberg equilibrium. So if you put them on the right-hand side, you can balance a ton before those water molecule hydrogen bonds will snap. And that's a good way to talk about cohesion and adhesion. And surface tension is a great one with dropping water on the penny. And then I obviously like to tie in all the different properties of life on Earth that are contributed to or by this different properties of water. So different things that I like to talk about. I always like to show them the graph of temperatures along the coast of California, just because it's a really good way to show that because we're near a massive body of water that has a very low specific heat, we can talk about specific heat and kind of the different nuances of high versus low specific heat, work on some graph analysis with those types of things, and then be able to show that since that water temperature is not changing much, that beautiful coast of California, especially San Francisco, gets very temperate climate year round. So that's kind of an interesting discussion. And then you can always tie in how cohesion and adhesion allow water to travel from the roots of the plant up to the leaves. So I like to talk about all of those things. And then the fact that ice floats, right? The fact that water is one of the only things on Earth where the solid is less dense than the liquid, hence ice floating, which allows for a thriving marine population. So those are just some of the things definitely to talk about that can kind of bring the structure of water and hydrogen bonding, which can be, if you're not a chemist at heart, a relatively dry, bland topic, more to life. Anyone have questions on structure of water and hydrogen bonding? Again, talking about the polarity. With water, it's always good to kind of highlight the difference between the covalent bonds that are between the O and the H in an individual water molecule, and then the hydrogen bonds between water molecules. That allows students to see that the same exact molecule can form multiple bond types. There's a lot of different things that you can incorporate in, but structure of water and hydrogen bonding can really be one class period with a few hands-on activities or diagrams to show these different properties, or some background reading. Okay, cool. We'll move on to next topic then. Okay, so elements of life is the next general topic. And again, this is just the pacing plan that College Board goes through. You can obviously kind of mix and match, but before you get into the macromolecules, you do need to talk about the elements that make them up. For me, I think because my students have a little bit of chemistry, I tend to go through this relatively quickly, maybe as an intro to a macromolecule day. I might not give it its own day. Because here we're really just introducing carbon, nitrogen, phosphorus, oxygen, and you can see an ENE. So if I highlight down here, let me just bounce back to the correct screen, get my pointer out. You know what's really annoying is if I'm using my pointer, this doesn't go away, and then you can't see the things that I'm trying to point at. Unless that does it. Oh, yep, it does. Okay. So carbon in all of our macromolecules, carbohydrates, proteins, lipids, and nucleic acids. It's kind of fun with elements of life. You can do some kind of pre-exploratory activities where they're looking at these different structures and categorizing what do all 4 of these things that look quite different have in common, and they should be able to point out the carbon, the hydrogen, and the oxygen that are in all of our macromolecules. And then you can start to highlight things like nitrogen and phosphorus in nucleic acids and proteins that kind of emphasize some of the differences and some of the nuances between our macromolecules, which will be fully introduced maybe that day or maybe the next day. And you can also kind of bring in here ionic and covalent bonding, and talking a little bit more about carbon if you want to start talking about the importance of organic chemistry and carbon and how it always makes 4 bonds and is an incredibly stable molecule, and that kind of gets kids thinking about why carbon is so important. I like to use my old organic chemistry kit from when I was in college to show them like the black ball that has the 4 indents in it for the 4 bonds that carbon can form, and they can kind of start manipulating with those kinds of things. I do a discussion-based classroom, so that's why there's a lot of these activities where students are kind of investigating some of these things. But elements of life as a whole of what College Board wants us to cover can really be implemented the same day that you're talking about macromolecules, especially if your kids have some sort of chemistry background. If they don't, then this might be the time to kind of pull out that periodic table, orient themselves to these things, talk about some of the key characteristics of carbon versus hydrogen versus oxygen. Good time to talk about the electronegativity of oxygen, because that becomes such a large thing to be discussed during the electron transport chain. So things like that. You can kind of touch on if you want to kind of dive a little bit deeper and give your kids a little bit more of an understanding. Okay, so then we can actually go ahead and introduce the macromolecules. And again, depending on if kids have had bio before, my kids haven't. My kids are also sophomores, so that's like another challenge in and of itself. But if your kids haven't had bio before, then this is going to be obviously a heavy hitter. But interestingly enough, so besides just kind of what we just talked about with elements of life, talking about carbon, hydrogen, oxygen, nitrogen, phosphorus, and those things in the actual structure, in this intro, a lot of what College Board wants us to cover is just the hydrolysis and dehydration synthesis reactions. So this is a key time to actually talk about how all of these things are forming. So this would be a good time to bring up monomer versus polymer, and what does mono mean? What does poly mean? How are these things coming together? What is hydro? What is lyse, right? Because they're going to see lysis, lysing, hydrolysis, lysosome, all the time. And then dehydration synthesis. What does it mean to be dehydrated? We're losing water. Synthesis. What does it mean to synthesize something to put together? So you can kind of go through and start the importance of Greek and Latin root words with students for AP, just because it's so important that they're able to identify things that they may not have been taught, because, well, you can't teach everything. There's just not enough time. And then it's also important in this one to kind of start talking about the types of bonds that are formed. So I always like to spend some time talking about peptide bonds and the structure of proteins and how those two are connected. Obviously, students may or may not be familiar with transcription and translation at the time, but we can still go over the structure of these things and how they're linked together before we get into all of the background of how that amino acid would even be coded for in the first place. So just kind of some food for thought there. Speaking of food, whenever we talk about the macromolecules, I always like students to brainstorm what foods have each of our macromolecules in them that we think about, which is just kind of fun and makes me hungry and students hungry, and a little off task, but kind of just gets them thinking about how these things are in the real world and why we consume them and what that means for us once we do. Any questions with the intro to biological macromolecules? Again, that doesn't have to be a full day either. Okay. So up next, now we're getting a little bit more dense. And what I found in looking through, reading and rereading the course and exam description guide time after time is that this first unit is relatively light on content, and again, is really just providing that background of chemistry to students. And you can also kind of take it a number of ways. So when we're looking at this properties of biological molecules, you'll see the essential knowledges listed are actually a little bit more dense for each of our 4 macromolecules, which is important. Definitely. But once we get into structure and function, it's going to get even more specific. And some of those things might be things that you want to save for a later unit, depending on when you're doing DNA replication, et cetera. You can kind of play around with it and place it in a couple different places, depending on what your plan looks like and how much time you have to discuss with students. So for properties of biological macromolecules, this is where we'll get to the actual structures of nucleic acids, proteins, carbohydrates, and lipids, but also why the different things that make them up matter. So for instance, in nucleic acids, it's important to start talking about the phosphorus groups, the phosphates and the phosphorus groups that are in the backbone, as well as the sugars that form up that backbone, and why that backbone, that sugar phosphate bonding along either side, is so much stronger than the hydrogen bonding between nucleotides, right? Because if they can start to become familiar with that, then they'll realize why the backbone hardly ever breaks, except for those little SNPs by topoisomerase, but why we can so easily cut through the hydrogen bonds that hold together our nucleotide pairs using helicase in order to replicate or transcribe. So those are the types of things that you should highlight, are just the chemical components that make up these structures are incredibly important to their overall function. And from teaching AP and taking, wow, my words, start of the school year, but from being to going to many conferences, structure equals function is such an important concept on the biology exam. And so the better that we can be with showing that, the better our students are going to be at finding those connections for themselves when they're in those application-based questions on the exam. For proteins, you can start talking about the different levels of primary, secondary, tertiary, and as well as the differences in the R groups and how that's going to have an impact on what that protein does and looks like and acts like, and how there's, our hydrophobic R groups, there's hydrophilic R groups, and what that means. You can start talking about the polarity of water again and that charge, and really talk about a lot of awesome chemistry within that properties of biological macromolecules. And then carbohydrates are just a good example of monomers versus polymers, and you can talk a little bit about the storage aspects of that, just because I think sometimes I personally talk about carbohydrates and then I talk about glucose pretty much solely, and I kind of forget to talk about cellulose and starch and some of those plant-based storage and structural elements. And then lipids is a good time to talk about really, really non-polar hydrophobic interactions. You can start introducing a phospholipid, just because that's going to be such a large component of obviously the plasma membrane, which is a huge part of our discussion once we get into cells and interactions within cells. There's 3, 4, or 5, I think, of the Unit 2 topics are all about cellular transport. And you can always do like a quick little example of oil in water, watching that kind of droplet come together, because I like to do that to talk about protocells as well, because I used to always teach evolution first, but it's kind of fun to talk about that and see those interactions, and then talk about why the phospholipid bilayer is the way that it is. Questions about that? Okay, so then this next part is kind of interesting. So the structure and function of biological macromolecules. If you take a look at what College Board has us teaching next, and again, this is obviously up to interpretation and construction by each individual teacher, but actually the structure and function goes into all the directionality and subcomponents of each of these macromolecules and how that influences kind of their structure and their function. So this is something that I've kind of been debating myself of how I'm going to incorporate it in teaching it to students. But a lot of this talks about like the directionality of DNA. That's something that sometimes, like, take it or leave it. They've incorporated it into Unit 1 because our next major topic is nucleic acids, and it's going to talk about the replication of DNA. So that's something that you can tackle, or you can save it, because a lot of teachers like to do that in a genetics unit, talk about the structure of DNA, the replication of DNA, and then transcription, translation, and all that good stuff. There's also I'm just going to look back at our course framework, because there's also a section, not science practices, but course content, of gene expression and regulation, where that might fit nicely as well. But you can see here, these essential knowledges are going to comprise kind of how the structure of that individual macromolecule influences its function, just to the next level of what we talked about last. going more into talking about like alpha helices and beta sheets for proteins and what that means and how that connects to a polypeptide and how that protein is functioning, the different chains that can form between carbohydrates, the fact that DNA is anti-parallel again and has directionality to it. So you can start talking about these things, and this is a really good opportunity to start bringing in some models and showing you could throw in enzymes here and talk about how the folding of that protein is going to lead to an active site, which is going to give enzymes specificity. So you can kind of incorporate some other biological things because enzymes haven't really been touched on yet as far as things that College Board wants us to cover, but would be something very, very important to highlight the structure and function of a protein since it's such a special protein. Also, I think just talking about the three-dimensional shape and the things or the fact that these things don't live in isolation or just live on the page or just look like N's, C's, H's, O's, P's, things like that, is important for students to see, and that there's a reason behind the folding patterns or the twisting patterns or the ladder formation or whatever it may be. It's not just some sort of random event. Any questions about that? This is also a good exploratory activity where you can talk about, okay, here's the structure of DNA. Tell me how you think that might contribute to the function, or here's the structure of a protein. How might that contribute to a function of that protein? You can show the linear chains of a carbohydrate and talk about how that might lead to enhanced storage in plants or in animals, and kind of go into some deeper explanations of how these things came to be. You can bring in some evolution there too, right? Like none of these structures and functions are random. They're all highly conserved and passed down from generation to generation. Okay, and then the last big thing to talk about is nucleic acids. So here it's just a general overview where you can talk about the differences between DNA and RNA and why that's so important. I always like to highlight, sometimes I think that we can kind of pigeonhole and be like DNA is double-stranded and RNA is single-stranded. DNA is mostly double-stranded, RNA is mostly single-stranded, but there's exceptions, and I like to bring up some kind of weird viruses and things like that, and kind of talking about how different organisms may use this differently than we do. Whew. I apologize. And you can go even more in depth. So the reason that nucleic acids is their own section here is just because the students do need a deeper understanding of the structure here in order to have a deeper understanding of the function, since it is such a large part of the heredity unit, the gene expression unit. Truly, it has so many implications. When we're talking about proteins, we're talking about things that were coded by nucleic acids. And you can talk about the directionality, again, the different sugar, the different bases, and start doing some basic pairing things. I also think this is a really great opportunity to throw in a lab, right? If you're just doing like a 5 to 7 day, maybe week and a half, or 2 week kind of unit, you could do a really cool exploration at the end from something as small as just the like strawberry DNA extraction or cheek cell DNA extraction. That's pretty easy to do and just allows students to see some DNA, to something that could be more complex. You can start bringing in some more complex lab skills and talking about, contamination and keeping things pure, and depending on if you have resources to get samples of DNA to do something a little more intense at the end of this unit. You can go into a little bit more depth. But most importantly, you're able to kind of just talk about why this is so important, get students really excited. I think this is like a really great end to the unit just because you can use it to get students really excited about their own genetics, talk about what the rest of the year is going to look like and how nucleic acids hold such a power over the individual no matter what, and kind of just like frame and then do a really fun activity to end it. But this unit is relatively short and relatively straightforward. As far as like units go, there's not a ton of misconceptions that I can think of that students have. A lot of it is more memorization based, just because they're going to need to know that having a nitrogen means that that thing is not a lipid or a carbohydrate. Having a phosphorus means that it's most likely a nucleic acid, things like that. Students just need to be familiar with, but the more you expose it to them, allow them to see models, allow them to kind of have hands-on experiences, the more that's going to naturally kind of happen for them. Any questions about the rest of Unit 1? Cool. If there are certain things that you want to go through on these teacher-led unit reviews, just let us know, because we didn't do these last year. We've just been focusing on student presentations. So if there are certain things that you want us to include before our next one, when we talk about Unit 2, just drop a comment or something like that, and we can be sure to include it, whether it's practice questions or a specific concept that you would like to go over. If there's ever something specific that you want to talk about, you can always chat it or do something like that, and we can make sure that we get that done for you. I'm sure everyone's wiped though. Teaching is hard. Getting back in teacher shape is really, really hard. The legs hurt, the brain hurts, the heart hurts, everything hurts. So if there's no other things, then I will just head out for now. And if you guys ever have questions, you can just let me know, and chat if there's something else that you want to include in the future, because this was the first teacher-focused broadcast, usually all student-focused broadcasts. So we can certainly tweak and add things that you guys would want to see. All right, have a great night, everyone.